Hello, everyone. Welcome to this webinar, a tutorial of FlowJo version 10. Just start my video here. And make sure we're recording. Looks like we are. My name's Ian Taylor, and I'm a director here at FlowJo, and I think I'm going to get started on this webinar. If you have any questions along the way, particularly general questions, feel free to throw those out in the questions pane associated with this Zoom meeting, and I'll get them answered along the way. I should be able to see your questions as you ask them. If you have specific questions, you might consider holding off and either asking at the end of the presentation or submitting them to me in my email or to techsupport@flowjo.com. And we'll get to those as soon as possible. All right, so without further ado, again, this is going to be kind of a general overview of all the major functions and features in FlowJo version 10. I'm not going to get into too many specifics here because this is an intro. And so for the beginners, people that haven't used FlowJo before, we want to give some kind of broad strokes and just get you familiar with the general pieces. So as I mentioned, the first section of this intro is going to be very general, just talking about how you navigate the workspace, customizing the ribbons, doing some graphing and gating, and dealing with the layout editor. And then we'll get into some more specifics on performing analysis in the software in the second part. So first of all, you're going to want to be familiar with the FlowJo workspace, which looks like the following. And it is composed of three general sections. The first is the ribbons and tabs area at the very top of the workspace. Next are the groups, where you're going to arrange your data files. And finally, the sample section, which will show the samples contained in all of your various groups. With regard to that first section, ribbons, tabs, and bands, it's going to be important just to understand some of the nomenclature here. The Applications button is the button on the far left, which when clicked will allow you access to the saving functionality and new workspace options. In the taskbar, you have some of the most commonly used tools, sort of generic tools within the software, such as add samples, create a group, hotkey for the compensation wizard, the table editor, layout editor, and the recalculate button. We've recently added functionality that connects FlowJo directly to another product, FlowJo Envoy, and the documentation for that is accessible through the button top right-hand side of the taskbar. The tabs contain different ribbons and bands within the workspace, and those bands are composed of different options that are similar. So for example, in the navigate band there, we see new workspace, add samples, create group, table editor, etc. And so those are going to help you do broad actions within the workspace. The ribbon configuration window is really important if you ever want to change or reconfigure those tabs and the ribbons that they contain. The preferences is going to be more important for changing the workflow preferences and defaults within FlowJo. In order to customize the ribbon, as I just mentioned, you'll click on that little ribbon configuration button, which will open up the ribbon configuration dialog, where you can drag the various actions into tabs within FlowJo's workspace. And in that way, you can actually customize the ribbons that are contained in every tab within the workspace. So if, for example, you wanted to see the biology band in the FlowJo tab, you would just drag that biology button in the top right-hand corner of the ribbon configuration window directly into the FlowJo tab, and you'll see it appear there. If you ever want to remove one of the bands from a tab, you can just drag and drop it from the workspace off onto your desktop or out into space, and it will remove it from that tab. Another super important feature within FlowJo that you'll want to know right off the bat is how to import data. I find the easiest way to do this is simply to drag and drop a folder containing your FCS data files right into an open FlowJo workspace. So that's shown in step 1, illustrated by dragging that folder from the right-hand side of the workspace directly into the area that says drag samples here. So that's a clue. Also, in the FlowJo tab, if you click Add Samples, it'll give you a couple of different options. One is just to choose Add Samples, which will open up a browser window where you can find and select the FCS files on your computer, and that will add them directly to the workspace. Alternatively, as I mentioned, we've now connected the FlowJo Envoy product directly to FlowJo. And if you click Add Samples from FlowJo Envoy, it will actually bring up an Envoy browser that allows you to add data from workflows that you saved in that product directly to FlowJo without having to download the files first. So those are the 3 methods of importing data. Next, we're going to talk about another really important area of the workspace, which is the groups pane. The groups act similar to the file folders in your computer's directory in that they organize the different files that are contained in the workspace. Now, one sample can be contained in multiple groups, so they don't act exactly as a folder would, where one file would have to exist only in one folder. Another really handy thing about these groups is they actually allow you to apply your analysis via a grouping action. So if you drag and drop a gate or a statistic that's been applied to one sample to a group, it will apply all of those functions to all of the samples contained within that group. Another thing you'll notice right away if you're using groups is whatever font style and color the group has applied to it will be applied to those nodes to the group. So here we see in the master gates group, all of the gates there are italicized and colored red, just like the master gates group. And if we were looking at an individual sample, you'd see that reflected as well. We'll see that more going through these slides. In order to create and edit groups, you can always click the Create Group button. There's also a hotkey, which is Command G or Apple G, as illustrated here. That will open up a Create Group dialog, which will look very similar to this Modify Group dialog. In order to modify a group, you just double-click on a group, and it'll open up this Modify Group dialog. Within, you can do things like name the group, change the role of the group, change sample inclusion criteria, which I'll get into a little later. You can even use it for advanced users. You can assign keywords via a group, which is really handy for some analyses. Now, speaking of sample inclusion criteria, this is a really handy function within groups, especially for template analyses. So what this is meant to do is to add samples to a group based on things like keywords. So if all of the samples that you want included in your first group have a well ID that begins with the letter A, you can set the keyword well ID to contain A as a sample inclusion criteria. And in that way, say the first row of your plate will be included in that group, assuming your first row is the A row. There's also the possibility to include include samples in a group based on their staining combination. All staining combinations for all samples added to your workspace will be available as sample inclusion criteria within the list that you see there under the sample inclusion criteria section. So here we have the HLADR, PERK1_2 blank CD3. Those are all of the stains that have been added to this workspace, for example. Samples and sample analysis. Here we're just talking about applying gates to a sample or to a group, which will then apply those gates to the samples. You can also add things like statistics, which can be group applied as well, and they will be calculated for each of the populations to which they've been applied within a given sample. In order to add gates, you're just going to double click on a sample to open up a graph window where you can manually draw a gate if you haven't done so already. Within the graph window, you'll see there are several different types of gates available in the gating tools section at the top of the graph window. You'll also see that there are various different plot view options and plot types that you can choose from. We'll go over the details there in a moment. You can also select your active gate and do things like change the gate into a not gate by unchecking the events inside option or changing the color of the gate. In this case, we've set the tint to red so that that CD3 positive gate appears in red. As far as gating tools, there are a variety. Here we're illustrating 3 of them. One that's not shown is the rectangle gate, and of course that just draws a rectangle around your population of interest, if it's amenable to that. There's also the quad gate, which is very popular. It allows you to essentially set one cutoff for X and Y axes with a single click. The ellipse gate is shown there on the left, and it's the 3rd gating tool in. The polygon gate is the next one, shown here almost in a rectangle fashion. And there's also a pencil tool that allows you to free form a gate if you prefer. We don't see that one being used a lot. And then there's the auto gate, which simply sets a gate based on contour lines. So it's also been called a density gate. One really handy thing, if you're using the auto gate and you find you've landed on the wrong contour, you can click the active gate option at the bottom of the graph window and either click the plus or minus sign to change the contour on which that auto gate has been applied. So that's a really important feature in FlowJo, obviously, gating. The different plot types I mentioned come in these flavors. You have the default pseudo-color plot, which is just a density color mapping, essentially. You have a contour plot, which can be set to show outliers, as it is here, top center. Density plot, zebra plot. You have the dot plot, very simple, probably the most simple of the 2D plots. And finally, the histogram, which is really should be called a 1D histogram, since all of the plots in FlowJo are technically histograms. They're just mostly 2-dimensional histograms. Another super important thing to be familiar with before you really get started with your analysis is the ability to change or customize your axis scaling and transforms. This can be done by clicking the T button next to either axis in the graph window and choosing Customize Axis, which will bring up the window on the right. Speaking of that transform window, there's a lot that can be done here. A sample workflow shown here is just to select the parameters for which you'd like to change the scaling in the menu on the right. And then to change the decades, you can click the plus minus signs next to either end of the axis. In the case of the bi-X transform, it's actually only the positive end of the axis that will allow you to adjust in that way. You can also add, remove negative decades or change the range manually in the area below where it says scale. Again, in the case of many of these transforms, you can use a width basis to adjust the R value associated with your transform. It's going to increase or decrease the area around 0 shown in a linear scaling, which is usually very handy for compensated fluorescent channels. That's why that transform was kind of invented. And before I move on, you'll also notice there's a few options at the bottom of that transform window. One is simply to apply the transform that you've changed to your data. When you do click apply, it's going to apply for all of the parameters chosen in a given compensation matrix. So in other words, every sample that contains to which that compensation matrix has been applied will receive those changes to the axis scaling. If you click save, that will actually pass on the changes to the transform to your preferences, so that the next time you open up a similar data set, meaning a data set from the same cytometer with the same parameters, those transforms that you've set will be applied automatically. So this is really handy if you want to change the sort of global default scaling of a set of parameters for your cytometer of interest. The effects of transformation are essentially aesthetic. They don't change the statistical values for your data outside of biological relevance, although there can be some changing based on the rebinning of your data. But mostly it's to make the data look sensible and so that it's available for analysis in the way that a researcher would like. Oftentimes people are trying to match their acquisition-defined display with that in FlowJo. And frequently this will not be the default, so you'll have to make some adjustments through that T button. If you ever have questions with this, we would encourage you to send over an example data file to techsupport@flowjo.com and include with that, if you can, a screenshot of how that data looked in your acquisition software, so we'll have something to compare against. Adding statistics is obviously a crucial aspect within FlowJo, and it's done by accessing the Tools tab first and clicking Add Statistic. Within that Add Statistic dialog, as you'll see, there's a list of available statistics which you can choose. Some of these statistics, like count, don't depend on a parameter, but some do. For example, if you're asking for a median, you have to also tell FlowJo which parameter you'd like that median calculated for. And you can select multiple parameters within that Add Statistic window. Prior to applying the statistic, you'll be required to select at least one population, but you can select multiple populations as well. And then when you click the Add button within the Add Statistics window, whatever statistics for whatever parameters you've selected will be applied to whatever populations you have selected. So this can become a little bit complicated, but it can also be done very efficiently once you get used to the platform. And just like any other node in FlowJo, you can always apply the statistics nodes to groups or group-owned gates in order to apply them to a set of samples, if you wish. The layout editor is the area where you're going to create graphical reports. So you're going to add populations from your workspace to the layouts in order to eventually publish them or export them for your PI. In order to add graphs, we normally recommend that you just drag and drop a population of interest from your workspace directly into the layout editor. Whatever set of parameters you were viewing for that given population are the parameters that will be added to the layout for this population by default, and you can always change that. We'll go over that a little bit more in detail as we go. To access the layout editor, you're just going to click the layout editor buttons available at the top of the workspace and also in the navigate ribbon. You can create new layouts by clicking the plus sign in the top left-hand corner of the layout editor tab of the layout editor. You can duplicate layouts in case there are plots that you want to make some adjustment to, but keep your original within that layout editor. Within the layout editor tab in the layout section just below the add or create layout button. And below that is a minus sign, which indicates it's going to delete whatever layout you have selected. So be careful with that. Layouts can be batched, and information on the iteration of that batching is available in the iteration section shown here. You can also, in the iteration section, modify what group is being viewed within the layout. In the batch section, you'll actually create your batch, and we'll go into a little more detail on that in a moment. Graphs can be organized and reformatted within the layout editor. This is being shown here where we're adding ancestry and adjunct histograms to the 2D plot that's currently been added to the layout editor. Again, just right-click on the plot and you'll have a whole variety of options, including ancestry, backgating, legend, annotation, etc. And that's how you can format or reformat the plots that you've currently added to your layout editor. Some of the basics of working with the layout editor, again, This window contains tabs, so layout editor, file, edit, object, and arrange. It also contains bands, which can be customized. This is done similar to the way that we illustrated for the workspace. Another super handy way to reformat plots within the layout editor is to just double click on a plot, which will bring up a graph definition window, which gives you a set of tabs. Within the specify tab, you'll see options to change the X and Y axes being utilized in a given plot. You'll also see various graph type options, including all of the graph types that we've showed previously, and also those options to show outliers, change the size of dots, and to do smoothing for things like pseudocolor or histograms. In the annotation tab, you'll have a lot of different annotations available, including the X and Y axis labels. So we see a lot of time the X and Y axis labels that people have imported from their acquisition software or their cytometer may be ambiguous. They might just say FL1 for your fluorescent channel 1, but you may want to relabel that to PE. Well, this is a great place to do that. You can just change the axes labels here to match whatever your desired axis label is. Batching analysis in the layout editor allows you to bring in similar plots from a group of samples within a new layout. So the batch options are all within layout editor tab by default, and they'll require that you specify the group over which you'd like to batch the plots. To select the iteration, usually sample is used, but there are other options for advanced users. And finally, sorry, not finally, but importantly, you're going to set the report type that you want batched within the batch section of that layout. In this case, we've chosen to batch to a web page. Oftentimes, you'll want to batch within FlowJo. In that case, you'll choose batch to display, and that will allow you to preview the batch that you've created before exporting. And you could still export as an HTML if you wanted to bring this in as a web page, for example. Finally, once you have all of those settings adjusted the way you like, you'll just click the Create Batch Report icon, which should be lit up the way it is in this picture, and that will generate a batched report. Sometimes this is what your PI will require once you've finished your analysis. Sometimes it might be what you want to send to your publisher to illustrate the analysis there. The table editor, similar to the layout editor, requires that you add populations or statistics to basically an area that previews the table that you're going to create when you batch. And that batch function is called create table in this case, but it's going to act very similarly. The iteration is going to be the same. You're going to choose which group you want to batch across. And to access the table editor, you're going to click the little table editor buttons back in the workspace under either navigate or in the top section of the workspace. Once you've got the table editor open, you can begin bringing in the statistics that you're going to want to use in your table. And this is done, again, similar to the way that you added populations within the layout editor. You're just going to drag and drop your populations or statistics directly into the table editor section. You'll see there that every column that will be created in the table, which we'll show in a moment, is going to be previewed as a row. So the very first one here is just a keyword. Star PID is going to be the first column of our eventual table. If you drag a population directly into the table editor, by default, the statistic you'll see is frequency of parent. So you'll see frequency of parent is listed there many times, but you can actually change that if you just double click on one of these rows to select a different statistic. Within the table editor, again, you have a set of tabs, the bands of which can be customized with the ribbon configuration window. And here we show just the default tables, iteration, and output. The table editor has the ability to create visualizations as well. If you click the Visualize tab, you can choose things like heat map or expected range. And this will actually make a heat map out of the statistics that are being shown in the resulting table. Here we see a table that's been batched and is showing various statistics heat mapped so that the low values are blue and the higher values are yellow, or become progressively more yellow. Once you've batched this table, in this case we've batched to display, you can go to the File tab and choose Save As, and then save the table in a variety of formats to your local machine. You can also batch directly to the layout in case you want to show a summary table for your plots. You can also batch directly to file if you don't want to have to deal with saving the file from your displayed table. All right, so in the second section of this tutorial, we're going to touch on a lot more of the analysis functions within FlowJo, compensation, exporting, concatenating, just touch briefly on the cell cycle analysis platform, plugins, templates, and then I'll show you some additional resources. So oftentimes, the first thing that you're going to want to do right after you've added your data to FlowJo's Workspace is to compensate. And probably most of you are aware that you have to compensate in multicolor flow experiments. This is because spillover between channels interferes with the information coming in from each detector and has to be corrected for typically. Here we're just showing the various spectra of dyes that are frequently used in flow and how they overlap with one another, showing how much how important it is to perform compensation in multi-flow data. Especially the more parameters you use, the more important this is. There are 3 general rules of compensation which are very important to keep in mind. One is that your single stain control for every parameter in the experiment must be defined. So you can't have compensation matrices that contain fewer controls than the parameters that you hope to compensate. Controls must be at least as bright or brighter than any sample for which the compensation will be applied. That's the positive control, obviously. The background fluorescence should be the same between each of these negative and positive control populations. And the compensation must match exactly the experimental fluorochromes in your data files. So even a slight renaming of the parameters will actually throw off FlowJo's compensation. So it's very important that your single stain control parameters match exactly what your experimental parameters are. In order to access the compensation wizard in FlowJo, there are 3 things you must do. One is you have to actually add compensation controls to a compensation group. In other words, that group's role must be compensation, and by default, there'll be a compensation group in a fresh workspace within FlowJo. Once those samples are added, you have to select the compensation group, and finally, just click the compensation tool, which appears in the cytometry section of the Tools tab within FlowJo's Workspace. And the button looks like this. It's 2 overlapping peaks showing some need for compensation there. And after you've clicked on that button, we actually have a wizard that will auto-gate your samples for you. And this can be very handy, but as we'll illustrate, it should be checked very carefully that the auto-gate was performed correctly for both size populations and also for the negative and positive control populations. So once you've clicked on the compensation wizard, you'll get this wizard dialog open, which illustrates a set of columns corresponding to the parameters that are contained in your data, the samples associated with each of those parameters, single stain control samples in this case, negative populations, negative control populations, and positive control populations. These are, again, all going to be filled in automatically by FlowJo's compensation wizard, but you should be very careful to check that every one of these columns is accurate, that the parameters you're hoping to compensate are listed, and none that you don't want to compensate are listed, that the samples assigned, the single stain control samples assigned to each parameter are correct, and that the negative and positive control populations are correct. If you need to adjust any of those, you can always, let me go back one, you can double click on any of the graphs in the bottom preview section of the compensation wizard in order to open up a graph window and adjust the gates manually. If you've done compensation yourself or you've redrawn a gate outside the compensation wizard, you can always change the sample or the population that's used for compensation by clicking on the drop-down menu there. So any of these samples, negative or positive control populations will be adjustable through a window that looks like the following, simply by clicking on that cell in the table. Once your compensation is finalized and can be calculated, or you're done with your compensation, you can click View Matrix to view the compensation matrix that's been generated based on the controls that you've chosen. You should be careful not to close the compensation wizard until you are satisfied with the matrix you've generated, in case you want to go back and adjust any of those controls. Once you've closed the wizard, FlowJo is not going to save those assignments. And if you reopen the wizard, it will actually it will perform the auto assignment again. And so sometimes that can be desirable if you just want to start from scratch, but frequently users can be frustrated by that. So just be careful that you're not closing the compensation wizard until you're done with your compensation. Once you've clicked View Matrix, you're going to see a window like this, which illustrates the compensation that's been calculated for each parameter into each detector. The detectors are shown as columns, and the rows themselves are the dye or the parameter being spilled into each of the detectors. Within this compensation editor window, you can change the color associated with the compensation matrix, which will appear in the grid to the left of samples for which that matrix has been applied. You can change the name of the matrix there. You can edit the matrix. Now, this isn't something that we necessarily recommend, but some users will want to go in and change these percentages for whatever reason. You can save a copy of your matrix. And a newly added feature is the Calculate SSM option, which will calculate for you a spillover spreading matrix for this compensation that you've done. It will only be available if you're looking at a compensation matrix generated in FlowJo. So for example, you'll see there's an acquisition defined matrix listed in this compensation editor, just above the compensation matrix selected on the left-hand side. And for that matrix, we wouldn't be able to calculate the spillover spreading matrix. This is because we don't actually know what control populations were used. FlowJo doesn't know what control populations were used by the acquisition software. You'll also notice that at the bottom of the compensation editor, there's a preview matrix, a preview of the matrix applied to a given sample, and you can change which sample you're previewing the matrix on so that you can kind of see how compensation is working and what the uncompensated data looked like relative to the compensated data. The uncompensated data will be like a turquoise green seen in this window, whereas the compensated channels will be black. Now, if you do click on the Calculate SSM button, or the SSM button, the spillover spreading matrix will look like the following. You can see here we've clicked on that button, and it's essentially going to show you into which parameters, or into which detectors, which parameters were contributing the most spillover. And in this way, you can optimize your panel design, and you can also check the quality of compensation over time. So this is a handy new feature that we've just implemented very recently. The effect of compensation, of course, is to zero out the negatives. So ideally, as you can see on the right, once compensated, your negative and positive control populations should have equal medians for the unstained channels. So in this case, we're looking at the compensation control for Q.605. And we can see here that the PE Texas red parameter has approximately equal negatives. Now, compensation is an approximation, so you'll virtually never get perfect compensation outside of a 3-color experiment. That is 3 colors or more, and you basically cannot get perfect compensation, but that is okay. Now, export and concatenate. This is another really important and sort of advanced feature within FlowJo. It allows you to just export particular populations of interest, or even to combine populations or samples of interest into a single data file outside of FlowJo, but these data files can be then re-imported into FlowJo or opened in another software, even exported as CSV files for import into something like Excel. In the case here, we see a titration experiment that's been concatenated, and we've included a keyword that allows us to separate out the different titer samples. And so we can see how the titration was affecting whatever color we're titrating for. In order to export or concatenate data, you're going to just select the samples or populations of interest in the sample area of the workspace below this menu, and then go to the File tab, and under Export Concatenate, choose either Export Concatenate Populations or Export Concatenate Group. I find it's much easier to just select the populations of interest and then use the Export Concatenate Populations in most cases. In the Export Concatenate option panel, you'll see a set of different options, one of which is the output. So as mentioned, you can use the FCS3 format if you want to re-import into a data analysis software for Flow, but you can also use the CSV export option if you want to import the data into something like Excel. And there are 2 different CSV output options. One is scale and the other is channel values. Scale values are corresponding to the fluorescence detected in your cytometer, and so typically that's the format that you would want to use. Sometimes people will want to see the data after it's been binned in FlowJo, in which case you would want to use the channel format, but that's much less common. You'll also want to note the destination where you are placing the exported or concatenated data file on your computer. Here we're exporting to the desktop. You'll also want to set the parameters that you're trying to export. In many cases, you'll want to use all uncompensated parameters. This doesn't mean that you'll lose compensation. If, for example, you're using an acquisition-defined compensation matrix, that will be saved in the keywords associated with your data file. So choosing all uncompensated parameters would still be appropriate and would still give you compensated values if you re-import into FlowJo. If you're doing compensation manually, as we illustrated previously, and you don't want to have to fiddle with exporting your matrix and re-importing your matrix into a new workspace, you may want to use all compensated parameters. In that case, if you check that option, when you export the data, all of the parameters will have a prefix fjcomp, indicating that they were compensated implicitly in FlowJo, and you won't be able to see the uncompensated parameters at all. So that has some pluses, but also some minuses. You can also choose a custom set of parameters. If you just want to export a few parameters, you can select them there. Under advanced options, when you're just simply exporting data, you'll be able to change the name of the data file that you're exporting, or the populations that you're exporting. With regard to concatenation, you'll likely want to select a set of populations before opening up the export concatenation dialog. And this is because the concatenation function works to combine multiple samples or populations together into a new data file. This can be really handy for things like t-SNE, T-distributed stochastic neighbor embedding, which is a clustering tool that requires that you have all of the information in one data file prior to performing the operation. And so, like I say, a lot of people that are doing the t-SNE algorithm will want to concatenate their samples prior to running that function within the plugins section of FlowJo's workspace. Once you've chosen the concatenate tab, most things are going to work just the same as an export. The big exception being there's an advanced option called additional parameters, which you'll see at the bottom of the window there on the right. And if you click choose there, you'll be able to select a parameter that will be transformed in the concatenated file, a keyword that will be transformed in the concatenated file into a parameter. So in that way, you could do things like the titration experiment that we looked at. You could enter the concentrations for your various samples as a keyword, and then turn those keywords into a parameter in the resulting concatenate. Again, if you have any questions on this, it can be a little bit complicated. Feel free to reach out to myself or to tech support. After clicking on add a keyword, you'll see a list of keywords like this is listed for whatever samples you have selected. And you can choose to preview the keywords from a given sample and to select those keywords for eventual inclusion in the concatenated data file. Again, here we're just separating out the different samples in an ABC size 7 titration experiment, and that's just kind of neat. Another sort of advanced feature within FlowJo is the cell cycle modeling platform, which just uses Gaussian peaks or Gaussian curves rather, to model the G1 and G2 peaks of cell cycle, assuming you have a cell cycle die run with your experiment. And in that way, it will allow you to try and mathematically deconvolute the information from each of these cell cycle peaks, and gives you some information based on whatever model you've chosen. There's both the popular Watson and also the Dean, Jett, and Fox et al. Models available within FlowJo. We also have a set of plugins, and the list of plugins is growing every day, or at least every month. To access the plugins within FlowJo, you'll find there's actually a few defaults. If you go to the Workspace tab, look in the Populations band, you can find the Plugins button there, which will include all of the plugins that you've added to your plugins folder currently. And if you'd like more information on either how to use plugins or the latest plugins that we've released, you can access the FlowJo Exchange by going to exchange.flowjo.com. The link is listed there at the bottom of this window. And again, we're constantly putting out new features and functionality via the plugins that we have available there. So pretty excited about that. Workspace templates are essentially a way of saving some of the analysis that you've done on a given data set in case you're doing repeated analyses of similar data sets in the future. And the way this works, it streamlines repetitive analyses for multiple runs, and basically you just have to go to the File tab, choose Save As, and export your workspace as a template. Now, in the resulting template, what you'll get is a workspace that contains no data and only contains the groups you've created, the analyses you've applied to those groups, and any layouts that you've started and tables that you've started. Whatever whatever layouts or tables that you've batched won't be included in the template, similar to these samples, because when you first load the data into FlowJo's template, it won't know which sample to start with in order to create the batch. So some of the fine-tuning of your gates for each individual sample, and also the production of those batched layouts and tables will need to be performed after adding the data there. If you have more questions or want some training resources, you can always go to our webinars page, which contains recordings from our past webinars, and this one will be posted shortly. In case you have questions or want to review what I've talked about today. We also have guided instructional courses on FlowJo University, which is just through flowjo.com. If you visit that site and go to the Learn tab, you'll reach this URL. We also have a daily dongle blog that contains lots of tips and tricks for how to utilize the software in the most efficient possible way, and to do some troubleshooting as well. If you have any questions, we're here to help. I would just recommend, as I've said before, write to techsupport@flowjo.com, especially if you have advanced questions or you're having any trouble with your analyses. We'll be more than happy to take a closer look. And with that, I would also recommend send any example data files that you can or analyses that you can, along with screenshots illustrating any of the issues that you're running into or questions that you might have. I would also encourage you to reach out to me if you have questions about the use of FlowJo or about this webinar. My email is there, taylor@flowjo.com. All right, so we breezed through those 60 slides pretty fast. I didn't see any questions come up, but this is essentially the end of my tutorial, so I'll just hang out for a moment. If anybody has any questions, I'd be more than happy to field those. And thank you very much for your time. Appreciate you joining the webinar. Be sure to grab the next webinar, which will be available. If I back up a couple slides here, you should be able to find the next webinar. So one of our viewers is asking, can you touch on overlays? And yes, I absolutely can. I might even go so far as to open up the software, make sure you guys can see my screen. Let's see. I'm going to share my desktop with you guys so I can just start up the software. And I'll actually go through an overlay just to show you how an overlay is created and maybe some of the options that are available for overlays. Here's a panel of data that we frequently use for demos. And if I open up the layout editor, to create an overlay, I'm simply going to drag and drop a couple of these samples right on top of each other. So there's a layout, and I've just added, by dragging and dropping, sample B6. If I now want to overlay B5 right on top of this plot, I can do so just by dragging and dropping. And this is possible for any set of populations. So it doesn't have to be the raw sample. It can be subpopulations. Once they've been overlaid, there's a few things you can do. You can make multigraph overlays. So I can show all parameters by the side scatter. If I choose that, ours is very small here. Make that a little bit bigger. So here we're seeing a multi-graph overlay, for example. So hopefully that kind of answers just a brief overview of how to do overlays, but if you have more questions, I would encourage you to send us an email. We also have another question coming in, the minimum number of cells required for a statistically significant experiment. I think bare minimum is maybe 100 events, assuming you've done good quality control and you're sure about the nature of those events. But obviously, the more data points you have, the better. And in some cases, for rare cell analysis, we've seen people use many fewer than 100 cells. But obviously, maybe one event is not something that you could consider rigorous in the statistical significance. So that's a great question. You should always use more events whenever possible for doing statistical tests. Another question, if I'm looking for GFP positive expression, do I need compensation as well? Basically for just one channel. That's a great question. If you're only detecting one channel, then you can't actually perform compensation in FlowJo. So if all that you have is GFP as a fluorescent parameter, then I don't think you need to worry about it. Once you start to have, multicolor experiments, then you'll certainly need to compensate. The good news is maybe if you have only 2 parameters, your compensation can theoretically be perfect. But anything above that will be kind of an estimation, and the estimation will get sloppier as you add more parameters to the data set. But for only one fluorescent channel, I don't think you need to worry about compensation. It's a great question. Yeah. Right. Those were all really good questions. I might just finish out by showing you these additional resources one more time in case people have got their pens out and they're writing them down or need to see areas where they can reach out with more questions. Again, feel free to write to me, I'm at taylor@flowjo.com, or tech support@flowjo.com is another great resource for any of your questions associated with FlowJo or with your Flow experiment. And again, thank you all for joining. Bye now.